Welcome back, ladies and gentlemen. So we are continuing our discussion on the major organelles in your eukaryotic cells. And today let's pick up with your mitochondria. And this one and your chloroplasts are going to be ones that come back when we get to cellular metabolism. So it's going to be really important for you to sort of start picking up on the major components of these organelles here so that when we get to that point later this year, it's a little bit easier to have that conversation about where certain processes are occurring. So, your mitochondria is the site for cellular respiration, or at least the bulk of cellular respiration. And this is where we generate that ATP from all sorts of molecules, sugars, fats, so on. Although we'll talk mainly when we get to cellular respiration about the sugar metabolism, we are able to break down fats, proteins, other molecules in your body in addition to those sugars to ultimately help produce that ATP. But regardless of what we're breaking down, we need oxygen to do it. So this is an oxygen dependent process. So what's going to be important for us when we get to that point is really being able to discuss the structure of the organelle because that's going to drive where certain processes happen. So it is a double membrane. It's got that smooth outer membrane and then this really highly folded internal membrane. And those high folds are going to be really important when we talk about your electron transport chain and why we can produce so much ATP out of cellular respiration. Those folds do have a special name. We call those cristae. Um, and then you have your internal matrix. Now, remember that these guys have, they also have an internal membrane space, which is also going to become important as well. What I was going to say is, though, remember that these guys, along with the chloroplasts, are part of that endosymbiotic theory, right? We think these guys used to be free-living prokaryotes, and some of the evidence to that is the fact that they do have their own ribosomes. And those ribosomes do look a little bit more like prokaryotic ribosomes as opposed to eukaryotic ones. And they also contain their own mitochondrial DNA. So there are a handful of genes that sit in your mitochondria and do get expressed that aren't actually part of your nuclear DNA. And keep in mind, all of your mitochondria came from your mother. So they are maternally inherited as opposed to your nuclear DNA, which would have come half and half from both parents, which is kind of interesting. Um, the big support to that endosymbiotic theory, besides the ribosomes and the DNA, is the fact that they had that double membrane. We think that that outer membrane probably came from the endocytosis process, I should say, of the bigger cell that ate it, right? So this would have basically been a remnant of a food vacuole. It never digested the internal prokaryote, and so we were left with that double membrane and it actually makes it much more efficient when it comes to doing cellular respiration, and we'll talk about why later this year. The other point I want to make while we're here, speaking of that, is that it is found in both plant and animal cells. Highlight that, star it, circle it, do whatever you need to do. You need to remember that mitochondria exist in all eukaryotic cells because a lot of students, especially as underclassmen, will think plant, chloroplast, photosynthesis. That's true, but then they make the jump to animal mitochondria, cellular respiration, and leave it at that. And when we talk about photosynthesis and cellular respiration, your plants do both processes. Your plants do the photosynthesis, but they're creating the sugars at the end of that process to then turn around and use those sugars in their mitochondria to make the ATP so that their cells have energy to do other work. So your plants do both cellular respiration and photosynthesis, and as a result have both chloroplasts and mitochondria. When we look at animal cells, they only contain the mitochondria, which means they need to obtain those sugars from another source. So that's the big distinction between producer and consumer, or autotroph and heterotroph. So just keep that in mind. Make sure you understand that those mitochondria exist in both plant and animal cells because the plants do both photosynthesis and cellular respiration. Okay, speaking of chloroplasts, these guys, again, are only found in plants, periodic, and we tend to lump in the structure of plant cells with them. They also exist in a certain version in cyanobacteria. Now, remember, bacteria don't have true organelles, but they will have infoldings very similar to chloroplasts to allow that cyanobacteria to do that photosynthetic process. Chloroplasts are just one type of organelle that is in a whole family of organelles called plastids. We won't dive real deep into them, but just know that there are other plastids that exist. And this is the site of protein synthesis, or excuse me, photosynthesis, I should say. And this is where we're going to convert that solar energy into chemical energy. So again, they've got a double membrane system as well, and they've got that really highly folded inner membrane here that we talk about as sort of discs. A single disc is called a thylakoid, and then if we talk an entire stack of discs, they call them a granum. So I like to think of them as pancakes versus a grand slam, or that you might get at IHOP. That's kind of how I always remembered it. So grand slam, granum is a whole stack of pancakes as opposed to the individual thylakoid being a single disc or a single pancake. And that chlorophyll is embedded within those thylakoid membranes. These guys also have ribosomes, also contain their own DNA, because they also fit within that endosymbiotic theory. So again, these are going to allow for both the light and dark reactions for photosynthesis. So we'll talk about what those mean when we get to that point. And again, just like the mitochondria, these guys are also able to express a small subset of genes outside of the nuclear information because they also contain their own set of DNA, which is kind of cool. Also means that there are a handful of disorders that are associated both with that chloroplastic DNA, if there's mutations there, as well as mitochondrial mutations as well. So we'll come across a few of those as we talk through some additional content this year too. Last thing I'm going to throw here while we're talking about these guys is an organelle that's probably new to you, and these are your peroxisomes. These tend to be very highly concentrated in places like your liver because they will perform some detoxification processes. They are single membrane bound components. They also contain enzymes similar to what we would see with a lysosome, but what they do is they contain enzymes that transfer hydrogens from various substrates to oxygen, and they end up producing literally hydrogen peroxide as a result of that. So some of the breakdowns of fatty acids that occur during respiration use that oxygen. Some of them, when we talk about drinking excessive alcohol, right, alcohols contain oxygens and hydrogens, so they transfer them to creating that hydrogen peroxide. We use this a lot with the catalase enzyme that we'll talk about a little bit later this year. So what it really does is it takes chemicals that come into your body that are presumably harmful, breaks them down. Problem is the byproduct that they produce, while less harmful than the original substance, hydrogen peroxide in higher concentrations is also toxic. We use it as an antiseptic, right? If you've ever gotten a cut and used hydrogen peroxide on it, you can hear that bubbling, and that bubbling is often a result of the catalase enzymes breaking that hydrogen peroxide down. But it's toxic to any sort of bacteria that might be present if that bacteria can't break it down. But if you use it for long enough and you leave it on your skin for long enough, you eventually use up that catalase enzyme so quickly before your body can replenish it, and you'll actually see white rings of dead tissue around that cut if you leave it there for too long. That's what ends up happening in people who are alcoholics, chronic alcoholics, where they are drinking so much alcohol that they're utilizing these peroxisomes and we're producing so much hydrogen peroxide in their liver that they can't break it down fast enough. And so it ends up causing all sorts of damage to that liver tissue and If this happens long term, then we talk about things like cirrhosis of the liver because you're just producing so much of that hydrogen peroxide that from these peroxisomes that your body can't keep up breaking it down, and it just destroys that liver tissue as a result.